Hello everyone, welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. Today's class is about Immunology Part 4 for BSc MLT third year students. So today we'll study about immunoassays, that is radioimmunoassay, RIA, and enzyme-linked immunosorbent assay, that is ELISA. So before we begin exploring the details of radioimmunoassays and enzyme-linked immunoassays, it is important to understand what immunoassays are and why they are so essential in modern biomedical science. An immunoassay is a biochemical test that exploits the specific binding between an antigen and its corresponding antibody to detect and measure substances in a biological sample. So, these fundamental antigen antibody interaction allows us to identify, quantify, and monitor molecules that are present even in extremely small concentration, such as hormones, enzymes, drugs, and microbial antigens. So, over the years, immunoassays have evolved from early manually intensive procedures into highly automated, sensitive, and specific laboratory techniques that are used daily in clinical, diagnostic, and research laboratories worldwide. So, I'll give a short introduction about RIA and ELISA assays. So, radioimmunoassays or RIA is one of the earliest and most sensitive immunoassay techniques ever developed. It was introduced in 1960 by Solomon Burson and Rosalind Yellow, whose pioneering work on insulin measurement in human plasma earned Rosalind Yellow the Nobel Prize in Physiology or Medicine in 1977. So, RIA is based on a competitive binding principle where a radioactively labeled antigen competes with an unlabeled antigen from the test sample for a limited number of antibody binding sites. ELISA is an enzyme-based immunoassay technique used for the detection and quantification of antigens or antibodies in a sample. Instead of a radioactive label, an enzyme such as horseradish peroxidase, that is HRP, or alkaline phosphatase, that is ALP, is attached to the antigen or antibody. So let's start with radioimmunoassay, that is RIA, principle behind this and uses. The radioimmunoassay RIA is one of the most sensitive and specific immunoassay techniques ever developed for the detection and quantification of minute amount of biological substances such as hormones, enzymes, antibodies, and antigens. It operates on the principle of antigen antibody binding, similar to other immunological assays, but distinguishes itself by utilizing radioactive label molecules as tracers to measure the concentration of target analytes. This technique was pioneered by Solomon Burson and Rosalind Yellow in 1960 during their investigation of insulin metabolism. The profound impact of their work led to Rosalind Yellow receiving the Nobel Prize in Physiology or Medicine in 1977, recognizing the monumental contribution of RIA to clinical science and diagnostic medicines. So, RIA can measure substances present in concentration as low as 0.001 mg per ml or even less, which underscores its exceptional analytical sensitivity. The technique became a cornerstone in endocrinology and clinical biochemistry, revolutionizing our ability to detect and monitor hormones and other biologically active compounds that occur in trace amounts in the bloodstream. Applications of radioimmunoassay. RIA has an exceptionally wide range of clinical and research applications. It remains fundamental in the measurement of various hormones such as insulin, thyroid hormones, that is T3, T4, TSH, cortisol, and human chorionic gonadotropin, that is HCG. These hormones are critical markers in assessing endocrine gland function, fertility status, and metabolic regulations. Beyond hormones, RIA is applied in the detection and quantification of enzymes, vitamins, and serum proteins, providing valuable insights into nutritional, metabolic, and pathological conditions. One of its significant clinical uses includes the detection of hepatitis B surface antigen, that is HBsAg, an important marker for hepatitis B infection, and drug level monitoring for therapeutic and toxicological studies. Its precision and sensitivity make RIA indispensable in disciplines such as clinical endocrinology, toxicology, and virology, where accurate quantification of biomolecules at very low concentration is required. Principle of RIA. The principle of RIA is founded on the concept of competitive binding between a radio-labeled antigen, that is Ag, and unlabeled antigen, that is Ag, for a limited number of specific antibody binding sites. Because the antibody can bind to only a fixed number of antigen molecules, the labeled and unlabeled antigens compete for these sites. During the reaction, both antigens have equal access to the antibody. Whichever antigen encounters the antibody first occupies the binding site. As a result, the amount of radioactivity bound to the antibody is inversely proportional to the concentration of unlabeled antigen concentration present in the sample. The greater the concentration of unlabeled antigen, the fewer radio-labeled antigens are bound. To quantify result, a standard curve is constructed using known concentrations of the antigen. The unknown sample's radioactivity is then compared to the standard curves, allowing accurate determination of antigen concentration. These competitive principle forms the basis of all RIA formats. Next is types of RIA. There are two main types of RIA based on the phase in which the antigen-antibody interaction occurs. First is soluble phase RIA. Second is solid phase RIA. Soluble phase RIA, the antigen-antibody reaction takes place entirely in a liquid medium. Both reactants remain in solution throughout the assay. This format was commonly used in the early applications of RIA, particularly in hormone assays. Solid phase RIA, in this variant, either the antigen or the antibody is immobilized on a solid support such as polystyrene beads or microtiter wells. Immobilization specifies subsequent steps such as washing away unbound materials and measuring bound radioactivity. So both the types follow the same competitive binding principle, but the solid format offers greater practicality, reproducibility, and ease of handling, which is why it became the preferred method in diagnostic laboratories. Next is soluble phase radioimmunoassays. So in the soluble phase RIA, the antigen is labeled with a gamma emitting isotope, most commonly 125 iodine, due to its ideal half-life and energy characteristics. The assay mixture in this, the assay mixture consists of three components, that is radio labeled antigen, the specific antibody, and the test sample containing unlabeled antigen. When these components are combined, The unlabeled antigen in the sample competes with the radio-labeled antigen for binding to the antibody. As the concentration of unlabeled antigen increases, it displaces the labeled antigen, leading to a decrease in the radioactivity bound to the antibody. This decrease is directly proportional to the concentration of antigen in the test sample. After equilibrium is achieved, bound and free antigen fractions are separated traditionally by precipitation or adsorption, and the radioactivity of one fraction is measured using a gamma counter. The result is compared against a standard calibration curve to quantify the antigen concentration. Next is solid phase radioimmunoassay. The solid phase RIA improve upon the soluble phase format by immobilizing either the antigen or antibody on a solid surface such as polystyrene tubes, PVC wells, or microplates. Immobilization facilitates easy separation of bound and free fractions through simple washing steps. This washing steps eliminating the need for complex chemical precipitation methods. These methods allow for smaller sample volumes and higher throughput, making it specially suitable for large-scale or mass screening programs. Detection is typically achieved using radio-labeled secondary antibodies or radio-labeled antigens depending on the assay configuration. Solid phase RIA offers several operational advantages, which is faster washing, reduced background noise, and enhanced reproducibility. Moreover, its format paved the way for the design of modern immunoassays such as ELISA, which replace radioisotopic tracers with enzyme-based detection systems. This solid phase radioimmunoassay, figure A, B, and C. So, figure A is showing SS antibody, B to SS antigen, and C competitive binding RIA. So this diagram illustrates the concept of solid phase radioimmunoassay in three variations, that is A, B, and C. In figure A, the solid surface, often a polystyrene well, is coated with a known antigen. When a serum sample containing antibody is added, the antibody binds to the immobilized antigen. A radio-labeled anti-immunoglobulin antibody is then introduced to detect the bound antibody. The amount of radioactivity measured corresponds to the antibody concentration. In figure B, the reverse setup is used to assess antigen concentration. The solid surface is coated with antibody and the patient sample containing antigen is added. A radio-labeled secondary antibody binds to the captured antigen, forming a measurable immune complex. Figure C, it represents a competitive binding RIA. Here labeled and unlabeled antigens compete for a limited number of antibody sites. As the concentration of unlabeled antigen increases, less radio-labeled antigens bind. Thus, the radioactivity measured is inversely proportional to the antigen's concentration. Next is solid phase RIA variations. Several variations of the solid phase RIA have been developed to suit specific analytical objectives. First is antigen coated wells and second is antibody coated wells. So in antigen coated wells, used primarily for the detection of antibodies in a patient's serum, here the known antigen is immobilized on the plate and radio-labeled anti-immunoglobulin, that is example anti-IgG, is used for detection. Here in antibody coated wells, it is employed for the detection of antigens. In this setup, the specific antibody is adsorbed on the solid surface and the patient's antigen competes with the radio-labeled antigen for binding. These formats are often performed in 96 well microtiter plates, which are compatible with automated handling systems. One notable application of the antibody coated solid phase RIA is in screening for hepatitis B surface antigen, that is HBsAg, where even microliter quantities of serum are sufficient for testing. So, very small sample volumes in mu L range is required for this solid phase RIA. Next is competitive binding, that is solid phase RIA. The competitive binding solid phase RIA is a refined version of the classical assay. In this method, the antibody is immobilized on a solid support. A mixture of radio-labeled antigen and unlabeled antigen from the patient sample is then added to the system. Both forms of the antigen compete for the limited antibody sites. Following incubation, the unbound material are removed through washing. The bound radioactivity remaining on the solid phase is then measured. The critical feature of this assay is that the amount of bound radioactivity decreases as the concentration of unlabeled antigen increases. Thus, there exists an inverse correlation between antigen concentration and measured radioactivity. Quantification is again achieved by comparison to a standard curve. This format provides high sensitivity and precise quantification, making it a preferred technique in analytical endocrinology for measuring low abundance hormones. Next is advantages of RIA. RIA offers several noteworthy advantages that contributed to its historical and practical significance. Extraordinary sensitivity, capable of detecting substances in the picogram to nanogram range, far beyond the detection limits of most other analytical methods. Next is highly specific. High specificity, the use of specific antigen antibody interaction ensures exceptional selectivity for the analyte of interest. Minimal sample requirement, only a small volume of biological fluid is necessary, making it suitable for pediatric and limited volume samples. Next is quantitative accuracy. The use of standard calibration curves allow precise quantification with excellent reproducibility. These attributes made RIA a revolutionary method for clinical diagnostics and research, providing insights into endocrinology, pharmacology, and immunopathology that were previously unattainable. Next is limitations of RIA. Despite its high sensitivity and accuracy, RIA has several practical and safety limitations that have restricted its modern usage. Radioactive hazards, that means the essay involving handling and disposal of radioactive isotopes such as iodine 125, which require stringent safety protocols and regulatory compliance. High cost and complexity. Equipment such as gamma counters, shielding facilities, and isotope storage areas entail substantial financial investment and maintenance. Limited shelf life of reagent. Radio label compound decay over time, reducing assay sensitivity and necessitating frequent reagent preparation. Waste disposal. Disposal of radioactive waste require adherence to environmental safety standards and specialized containment procedures. Due to these challenges, RIA has been progressively replaced by non-radioactive immunoassays that offer comparable sensitivity without radiation hazards such as enzyme-linked immunosorbent assays, that is ELISA, and fluorescent immunoassays, that is FIA, fluorescent immunoassay. So together, limitation of RIA, it requires handling of radioactive isotope, disposal and safety concern, high cost of equipment and reagents, and gradually replaced by non-radioactive method, that is ELISA. Next is a replacement of RIA by ELISA. Modern immunoassays such as ELISA, enzyme-based, and FIA, that is fluorescent immuno-based, have effectively replaced by RIA. In these methods, the radioactive label is substituted with enzymes or fluorophores which generate measurable color or light signals instead of radiation. These newer methods are safer, cheaper, and easier to perform, and they eliminate radioactive waste management issues entirely. Furthermore, they can achieve equal or even greater sensitivity through signal amplification methods such as chemiluminescence or biotin evidence systems. Thus, the evaluation from RIA to ELISA represent a major advancement in immunodiagnostics, combining sensitivity with safety and efficiency. Next part is ELISA, sorry, EIA, that is Introduction to Enzyme Immunoassay. The enzyme immunoassay EIA is a method that uses enzyme label antigens or antibodies to detect the presence of specific antigen antibody reaction. It was first introduced in 1966 to localize antigens in tissue sections, providing an alternative to fluorescent labeling techniques. Later in 1971, enzyme-labeled reagents were adapted for serological testing, making the assay suitable for blood and serum samples. While EIA is widely used, highly sensitive and specific, can detect low concentration of antigen or antibody. Safe, no radioactive isotopes involved, unlike RIA. It is simple, versatile, and cost effective. That means it does not require complex instrumentation. It detects antigen antibodies or haptens, broad application in diagnostic and research. So it highlights that EIA was a major advancement over RIA in terms of safety and practical use. Next is principle of enzyme immunoassay. EIA works on the principle of enzyme labeling. The enzyme is attached to either the antigen or the antibody. When the substrate specific to the enzyme is added, a chemical reaction occurs producing a color change. The intensity of the color is directly proportional to the amount of antigen or antibody present in the sample. Commonly used enzymes are horseradish peroxidase HRP. It is widely used to stability and strong signal. Second is alkaline phosphatase, that is ALP. It is suitable for colorimetric detection. Third is beta galactosidase. It is occasionally used for specialized assay. So, principle behind this EIA, here the enzyme act as a marker or label which is attached to the antigen or antibody. When the enzyme substrate is added, it produces a color change which indicate a positive reaction. The intensity of color is directly proportional to the amount of antigen or antibody in the sample. Commonly used enzymes are horseradish peroxidase, alkaline phosphatase, and beta galactosidase. Next is types of enzyme immunoassays. that is homogeneous versus heterogeneous. So in this table, it compares homogeneous and heterogeneous enzyme immunoassays. In homogeneous EIA, all reactants, that is antigen, enzyme labeled antibody, and substrate are mixed together in a single phase. There is no separation step, making the assay rapid and simple. The reaction between antigen and antibody directly influences enzyme activity, altering the measurable signal. In heterogeneous EIA, one reactant, either antigen or antibody, is immobilized on a solid phase. After the antigen antibody reaction occurs, unbound components are washed away and the enzyme substrate reaction generates a measurable color. The ELISA is a classic example of heterogeneous EIA. Although this method involves multiple steps, it provides higher specificity and quantitative accuracy than homogeneous assay. So homogeneous EIA, that means single step assay, rapid and simple. Example is enzyme multiplied immunoassay technique, that is EMIT. Heterogeneous EIA, multi-step assay including washing steps to separate bound from free fractions, more specific due to removal of unbound components. So example is ELISA. Next is enzyme-linked immunosorbent assay, that is ELISA. So ELISA is a type of heterogeneous EIA where antigen or antibody is adsorbed onto a solid surface, typically a microtiter plate. The plate serves as an immunosorbent capturing one reactant. Detection occurs through enzyme substrate color reaction. Advantages: easy interpretation, that is clear positive or clear negative result, quantitative or qualitative assessment possible, high throughput capability using 96 deep well plates, ideal for clinical and research labs. So ELISA, it is nothing but enzyme-linked immunosorbent assay. It involves absorbing antigen or antibody to a solid surface that is microtiter plate. The plate will act as an immunosorbent, that means it captures one reactant, and visualizing it is using enzyme substrate color reaction. There are three advantages of ELISA. It is easy to interpret. It will give clear either positive or negative result. It is quantitative or qualitative based, high throughput using 96 Dipel plates. Types of ELISA. ELISA has several formats, that is direct, sandwich ELISA, indirect ELISA. Direct ELISA detects antigen using antibody-coated wells. Indirect ELISA detects antibody using antigen-coated wells. Other variations, that is competitive ELISA, it measures antigen concentration based on competition with labeled antigen. Capture ELISA, it uses two antibodies, one for capture and one for detection. Third is cassette, that is TRIDOT or rapid ELISA, point of care disposable test. So homogeneous is equals to rapid, heterogeneous is equals to more specific. Choice depend on application, sample type and required sensitivity. We'll see both direct and indirect ELISA one by one. Direct, that is sandwich ELISA. So purpose is to detect and quantify antigen in patient sample. Sample addition. So steps here, coating the specific antibody is adsorbed onto microtiter plate wells. Sample addition, patient sample binds the coated antibody. Enzyme conjugated antibody, secondary antibody binds to the same antigen and forms sandwich, that is complex. Washing, removes unbound antibodies. Substrate addition. Enzyme reacts with substrate that gives color change, indicates antigen presence. Interpretation. Color change, it means positive, that is antigen is present. No color change, it means negative, no antigen is present. Next is indirect ELISA. Purpose is to detect and quantify antibodies in serum. Steps: coating, known antigen adsorbed on microtiter wells. Serum antigen, patient's antibodies, if present, bind the antigen. Enzyme labeled anti-human globulin, bind patient's antibodies. And substrate addition, enzyme act on substrate, it will develop color. So interpretation is, if color appears, that means specific antibody is present. No color appears, that means antibody is absent, it means negative results. Diagram of enzyme link immunosorbent assays. It is divided into two parts, A and B. A is positive direct ELISA to detect antigen. B is positive indirect ELISA to detect antibody. So this figure demonstrates these two fundamental forms of ELISA. In figure A, direct ELISA, wells of microtiter plate are coated with specific antibody. The patient sample containing antigen is added, allowing antigen binding. A second enzyme labeled antibody specific for the same antigen is then introduced, forming an antibody antigen enzyme linked antibody sandwich. When the enzyme substrate is added, a color change occurs. The intensity of color is proportional to the antigen concentration in the sample. In figure B, that is indirect ELISA, the well is coated with a known antigen and the patient's serum is added. If specific antibodies are present, they bind to the antigen. Next, an enzyme labeled anti-human immunoglobulin, that is secondary antibody, is added which binds to the patient's antibody. Upon addition of substrate, a color reaction develops. This method is used for detection of antibodies in diseases such as HIV or hepatitis B. Next is competitive ELISA. Purpose is to quantitatively measurement of antigen concentration. Steps: known antibody mixed with patient's antigen sample. It will give some antibodies. It will bind antigen in solution. Mixture added to plate coated with same antigen. Only free antibodies bind plate antigen. Secondary enzyme labeled antibody binds primarily antibody and substrate is added which is then color measured. Interpretation: more antigen in sample, less freely antibody binds, that means weaker color. Less antigen in sample, more antibodies will bind, it means stronger color. So this figure of competitive enzyme linked immunosorbent assay, here the competitive ELISA diagram depicts a situation where an antigen in the test sample competes with plate-bound antigen for a limited number of antibody binding sites. Initially, a fixed amount of enzyme labeled antibody is mixed with the test sample containing unknown antigen concentration. This mixture is then added to wells coated with same antigen. If large amount of antigen is present in the sample, fewer antibodies will bind to the place-bound antigen, resulting in a weaker color reaction. When substrate is added, conversely, if little antigen is present in the sample, more enzyme-labeled antibodies bind to the plate, producing a stronger color signal. Thus, color intensity is inversely proportional to the antigen concentration. This type of ELISA is especially useful for quantitative assay of hormones and small molecules. So this competitive ELISA, it is an inhibition type assay where the concentration of antigen is inversely proportional to the color produced. Next is advantages of ELISA. It is high sensitivity and specificity, simultaneous testing of multiple samples, that is high throughput, quantitative result possible, and no radiation hazard unlike RIA. It is simple and cost effective, automatable, suitable for mass screening. So these are the advantages of ELISA. High sensitivity and specificity. It can test many samples simultaneously. High throughput. Quantitative result is possible. No radiation unlike RIA. It is simple and cost effective, and it can be automated for mass screening. Next is rapid cassette ELISA, that is TRIDOT method. So it is designed for point of care testing. Disposable plastic cassettes or dipsticks. It will give result in 5 to 10 minutes. No ELISA reader or wash is required for this. It is built in positive and negative controls. Application for this, it is used in HIV, hepatitis, pregnancy, that is HCG test. It can be used. Next is enzyme-linked immunospot assay, ELISpot. ELISpot detects and counts individual cells secreting specific antibodies or cytokines. Each secreting cell forms a colored spot where it releases its antibody or cytokine. The number of spot correlates with the number of secreting cells, providing a highly sensitive quantitative measures. Clinical application, it is useful in vaccine research, immunology studies, and monitoring immune response. This is the diagram of enzyme-linked immunospot, that is ELISpot assay. It is divided into two parts. A, it form photographic courtesy of R. Hutching and Blackwell Scientific. B, it is a schematic representation of ELISpot. So this ELISpot diagram demonstrates detection of individual cells secreting specific antibodies or cytokines. In figure A, an antigen-coated plate is used to capture antibodies secreted by B cells from a lymphocyte suspension. Each B cell that secretes a specific antibody leaves a localized color spot on the plate when treated with an enzyme labeled secondary antibody and substrate. In B figure, it shows a schematic of the same principle where anti-cytokine antibodies are coated on the plate to capture cytokines secreted by T cells. Each cytokine producing cell creates a visible colored spot after enzymatic reaction. The number of spots corresponds to the number of active secreting cells, providing a sensitive quantitative measures of immune response. ELISpot is widely used in vaccine research and immune monitoring. Next is advantages of ELISpot. It detects single cells producing antibodies or cytokines. It is highly sensitive and quantitative, valuable in vaccine studies, autoimmune disease research, and immune monitoring. So these are the three advantages. It detects single cells producing antibodies or cytokines. It is highly sensitive and quantitative, and it is useful for vaccine studies, autoimmune disorders, and immune monitoring. Next is biotin avidin enhanced immunoassay. So principle behind this, it enhances EIA sensitivity using biotin and avidin, which bind strongly. Steps involved: secondary antibody labeled with biotin, wells incubated with enzyme labeled avidin, which binds biotin tightly, and substrate is added, which enhance color reaction. Advantages: it detects extremely small antigen quantities. It is used in immunofluorescence assays, that is IFA as well. It provides signal amplification and high specificity. So this diagram, biotin evidin enhanced ELISA sandwich assay, figure A and figure B. Figure A represent monoclonal antibody, that is MAB, coated on the well of the microtiter well, reacts with test antigen, a specific antibody labeled with biotin. Figure B represent added to form a sandwich. B enzyme labeled avidin A is added. The binding of avidin is monitored by the conversion of the substrate to a colored product. So in figure a monoclonal antibody is coated on the microtiter well wall. The test sample containing the antigen is added, allowing antigen binding. Then a biotin labeled secondary antibody is introduced, forming a sandwich complex, which is capture antibody antigen biotinylated antibody. In figure B, enzyme labeled avidin is added, which binds strongly to the biotin moiety. When the substrate is added, the enzyme catalyzes the reaction, producing an intense color that indicates antigen presence. Because avidin binds biotin with extraordinary affinity, this system amplifies the detection signal, allowing measurement of extremely small antigen quantities. So these two are the diagram of biotin evidin enhanced ELISA sandwich assay. Next is application of EIA and ELISA. Clinical diagnosis, that is HIV, HBV, HCV, dengue, and COVID-19. Hormone assays, HCG, thyroid hormones, allergy testing, and autoimmune disease testing. Cytokine quantification for research, vaccine research, and immunology, monitoring immune responses. ELISA and its variants remain cornerstones in modern immunology and diagnostics, replacing older radioactive methods while providing high sensitivity, specificity, and safety. So this table is divided into three parts, which is technique, detects, and its key features. Direct ELISA detects antigen, and its key feature is it uses sandwich method and uses enzyme labeled secondary antibodies. In indirect ELISA, it detects antibody. It uses enzyme labeled anti-human globulin. In competitive ELISA, it detects antigen and it gives inverse color concentration relationship. In ELISpot, it detects secreting cells, and its spots indicate individual cytokine or antibody secreting cells. In biotin evidin ELISA, it detects antigen and enhanced sensitivity using biotin evidin linkage. So these three, part of table will explain a summary of these techniques and what it detects and its key features. Next is chemiluminescence. Chemiluminescence refers to the emission of light during a chemical reaction. In immunoassays, this property is exploited as a highly sensitive detection method. Unlike ELISA, which relies on colorimetric changes, chemiluminescence provides light output proportional to antigen or antibody concentration, which can be measured with a luminometer. Key advantages include high sensitivity. It detects very low concentration of analytes, low background interference. Unlike colorimetric methods, color substances in sample do not interfere, and rapid detection. Its results can be obtained more quickly than conventional ELISA. Chemiluminescence is widely used in automated immunoassay analyzers for hormones, tumor markers, and viral antigens. Principle of chemiluminescence assays. The principle involves a luxogenic, that is light generating substrate that reacts with an enzyme label. An enzyme such as horseradish peroxidase or alkaline phosphatase ALP catalyzes a chemical reaction. The reaction produces visible light photons proportional to the amount of bound antigen or antibody. The light intensity is captured by a luminometer and converted into quantitative data. Example of enzyme substrate system include horseradish peroxidase with luminol, and it is widely used due to high light output. Next is alkaline phosphatase ALP with deoxyetane phosphate. It produces stable luminescent signal. So chemiluminescence offers greater sensitivity and dynamic range than conventional ELISA, making it ideal for detecting low abundance targets. Next is advantages of chemiluminescence. Key advantages of chemiluminescence immunoassay include extremely high sensitivity, can detect minute amounts of antigen or antibody, rapid assay completion, fast kinetics allow shorter incubation times, high signal to noise ratio, minimal interference from colored biological matrices, quantitative measurement, automated luminometers provide precise quantitation, automation friendly, widely used in clinical chemistry analyzers for large scale testing. So applications include hormone analysis, tumor markers, viral antigen detection, and high throughput clinical diagnostics. So one in whole, advantages of chemiluminescence, it is extremely sensitive, it is rapid, no background interference will be seen, and it is quantitative measurement. And it is widely used in automated immunoassay analyzers. Next is immune blotting, that is Western blot. Western blot, also known as immune blotting, is a powerful method for detecting specific proteins in complex mixtures. It combines electrophoresis for protein separation and antigen-antibody reactions for detection. Western blot is most commonly used for confirmation of HIV infection and detection of viral proteins such as HBsAg. The method provides both qualitative identification and molecular weight estimation of target proteins. Principle of Western blot. The principle of Western blotting involves several sequential steps. First, proteins in a sample are separated according to their molecular weight using SDS page electrophoresis. Following separation, the proteins are transferred or blotted onto a nitrocellulose or PVDF membrane, which provides a solid surface for antibody binding. The membrane is then incubated with patient serum, which may contain antibodies targeting specific viral or cellular protein. If such antibodies are present, they bind to the corresponding proteins on the membrane. Bound antibodies are detected using enzyme-labeled secondary antibodies, and the addition of a substrate produces colored bands at the site of antibody binding. The presence and pattern of these bands allow for the identification of specific proteins or confirmation of infection. In this diagram of Western blotting test for HIV, figure A, B, and C, so the process of Western blotting can be visualized in three stages. First, electrophoresis separate viral or cellular proteins based on size. Second, these proteins are blotted onto a membrane and incubated with a patient's serum, allowing antibodies to bind to the target proteins. Finally, enzyme-labeled secondary antibodies are added, and a substrate reaction produces colored bands where antibodies have bound. The presence of specific bands indicates that the patient's serum contains antibodies against those antigens confirming infection. In the case of HIV, typical diagnostic bands include P24, GP41, and GP120. And the presence confirms a true positive result, reducing false positive from ELISA screening. Applications of Western blot. Western blotting has a wide range of application. In clinical diagnostics, it is primarily used for the confirmation of HIV infection, but is also applied for detection of hepatitis B protein and other viral antigens. In research and autoimmune studies, Western blot can identify specific antibodies against target proteins. Moreover, in molecular biology, it allows the study of protein expression patterns, post-translational modifications, and interactions. Its high specificity and reliability make it an essential tool for both clinical laboratories and research institutions. Flow cytometry. Flow cytometry is a laser-based cell analysis technique used to evaluate both the physical and chemical properties of cell in suspension. This technique enables a quantitative version of the fluorescent antibody technique, allowing researchers to measure cell surface markers with high precision. An advanced form, fluorescent activated cell sorting, that is FACS, allows not only analysis but also sorting of specific cell populations based on fluorescent intensity. Flow cytometry has become a cornerstone in immunology, hematology, and clinical diagnostics because it can rapidly analyze thousands of cells individually. Principle of flow cytometry. The principle of flow cytometry relies on fluorescent labeling of specific cellular antigens. Cells are stained with fluorescent label antibodies, then passed single file through a laser beam in a fluid stream. When the laser strikes each cell, light scatter provides information about cell size and internal complexity, while fluorescent light emission indicates the presence of specific antigens. Detectors capture these signals and electronics convert them into graphs, histograms, or numerical data. This allows quantitative and qualitative analysis of cell populations, making flow cytometry a versatile tool for both research and clinical applications. This diagram of flow cytometry, fluorescent activated cell sorting, that is FACS. This diagram represents the working principle of fluorescent activated cell sorting, that is FACS, and advanced application of flow cytometry. Cell labeled with fluorescent antibodies are carried in a fluid stream and pass single file through a laser beam. As the laser light interacts with each cell, detectors measure light scatter and fluorescence emission. When a target cell with a desired fluorescent characteristic is identified, the instrument charges that droplet electrically. The charged droplets are then deflected by an electrostatic field into separate collection tubes. This enables isolation of specific cell populations for further analysis or cultures. Flow cytometry components. A flow cytometer consists of several critical components. The fluidic systems ensures that cells pass in a narrow stream. This system ensures that cell pass in a narrow stream, allowing single cell analysis. The laser light source excites fluorescent labels attached to antibodies. Optics and detectors measures light scatter and fluorescent emission, while the electronics and computer system process these signals into interpretable data. The integration of these components enables high throughput, precise analysis of thousands of individuals cell per second. Next is flow cytometry application. So flow cytometry has numerous clinical and research applications. Clinically, it is used for immunophenotyping of lymphocytes, that is such as CD4 and CD8 counts in HIV patients, and for the diagnosis of leukemia and lymphoma by identifying abnormal cell markers. It is also used to measure apoptosis, cell cycle, and DNA content, and in research, FACS allows sorting of specific cell populations for further functional studies. Its ability to provide rapid, quantitative, and multi-parameter analysis makes it indispensable in modern laboratories. Next is immunoelectron microscopy. Immunoelectron microscopy combines the specificity of immunology with the high resolution imaging of electron microscopy. This technique allows detect visualization of antigen-antibody interactions at the ultrastructural level. It is particularly useful for studying viral particles, providing detailed information on the interaction between viruses and antibodies, which is not achievable with light microscopy. Immunoelectron microscopy principle. The principle involves mixing viral particles with specific antisera. Under the electron microscope, the viral particles forms clumps or aggregates where antibodies bind, making them visually detectable. This method has been applied to study viruses such as hepatitis A virus, rotavirus, and other enteric viruses. By directly observing the interaction, researchers can map virus antibody complexes and study their morphology at the ultrastructural level. Next is immunoferritin test. The immunoferritin test uses ferritin labeled antibodies. Ferritin, being an electron dense protein, is easily visible under an electron microscope, marking the exact location of the antigen. This technique allows precise location of viral antigen in cells and tissues and helps map the distribution of antigens within cellular structures. Its high resolution makes it an invaluable tool for virology research. Next is immunoenzyme test. In the immunoenzyme test, antibodies are conjugated with enzymes such as horseradish peroxidase (HRP). Tissue sections containing target antigens are incubated with this enzyme labeled antibodies. When a substrate is added, a visible colored product forms at the antigen site, which can be visualized under a light or electron microscope. This method allows histopathological localization of antigen and is widely used in both research and clinical diagnosis. So common enzyme is horseradish peroxidase. It is used for histopathological localization of viral or microbial antigen, research visualization of antigenic sites in cells and tissues. So summary of all these techniques: chemiluminescence, Western blot, flow cytometry, and immunoelectron techniques each rely on specific detection principle. Chemiluminescence uses light emission. Western blot separates proteins and visualizes antibody binding. Flow cytometry quantifies fluorescently labeled cells, and immunoelectron methods visualize antigen at high resolution. These methods quantitative, qualitative, and ultrastructural information, making them essential tool in clinical diagnostic and research. Last is key advantages of advanced technique. These advanced immunological techniques are highly sensitive and specific, enabling detection of very low antigen or antibody levels. They allow for rapid and automated analysis, provide both quantitative and qualitative information, and are widely used in clinical diagnostics, immunology, and research laboratories. Their development has greatly enhanced the accuracy, reliability, and depth of immunological investigation. So summary of today's class: general concept. Immunoassays are analytical method based on specific antigen-antibody interaction used to detect and quantify biological substances with high sensitivity and specificity. The earliest and most sensitive immunoassay was the radioimmunoassay RIA. It is developed by Solomon Berson and Rosalind Yellow in 1960. It uses radio-labeled antigens, that is AG, competing with unlabeled antigens for limited antibody binding sites. The bound radioactivity is inversely proportional to the antigen concentration or in the sample. RIA is widely used to measure hormones such as insulin, cortisol, thyroid hormones, viral antigen, HB, SAG, and drugs, though its use has declined due to radioactive hazard and disposal issues. To overcome this limitation, enzyme immunoassay, that is EIA, and its most popular form, enzyme-linked immunosorbent assay, ELISA, were developed. In this technique, enzyme replace radioactive labels. The enzyme acts on a substrate to produce a color chain, and the intensity of the color is directly proportional to the antigen or antibody concentration. So advantages of this are it is non-radioactive and safe, economical and simple, high throughput, and suitable for mass screening. Types of ELISA. Direct sandwich ELISA detects antigens. Indirect ELISA detects antibodies. Competitive ELISA measures antigen concentration based on color intensity, that is inversely proportional. Next is ELISpot. It detects antibody or cytokine secreting individual cells. Biotin Avidin enhanced assay increases sensitivity through biotin avidin binding. ELISA, it is safe, economical, and ideal for mass screening of diseases like HIV, HBV, HCV, dengue, and COVID-19. Hormone assays, allergy and autoimmune testing, cytokine quantification, and vaccine research. Chemiluminescence immunoassay represent a more advanced method using light emission instead of color change. Enzymes such as horseradish, that is horseradish peroxidase, HRP, or alkaline phosphatase, ALP, act on luxogenic substrate, example luminol, producing light measured by a luminometer. It is highly sensitive, free from color interference, and widely used in automated analyzers for hormone and tumor marker detections. Western blotting. Western blotting, immune blot, combines electrophoresis and antigen body reaction to identify specific proteins. After SDS page separation, proteins are transferred onto a nitrocellulose or PVDF membrane probed with patient serum and visualized as color bands using enzyme labeled secondary antibodies. It is the confirmatory test for HIV and useful in research on protein expressions. Next is flow cytometry, a laser-based analytical method. It measures all cell size, complexity, and fluorescence using fluorochrome labeled antibodies. Its advanced version, fluorescent activated cell sorter, that is FACS. This FACS allows sorting of specific cell types. Flow cytometry is widely applied in HIV monitoring, that is CD4 and CD8 counts, leukemia diagnosis, and cell cycle studies. So immunoelectron microscopy, that is IEM, it combines immunology and electron microscopy for ultrastructural visualization of antigen and antibody complexes. Its variants is immunoferrin test, which uses ferritin labeled antibodies, and immunoenzyme test, it uses enzyme labeled antibodies. Applications for viral identification, example is hepatitis A and rotavirus. For antigen localization in cells and tissues. So overall significance of immunoassays have evolved from radioactive, that is RIA, to enzyme based, that is ELISA, to fluorescent and luminescent techniques. They provide high sensitivity, specificity, safety, and automation. They play a central role in clinical diagnostic, immunology, toxicology, and biomedical research. Next is question parts. For question 3 marks, first define radioimmunoassay, that is RIA, and states its significance. Second, who developed RIA and what is its main principle? Third, mention two clinical applications of RIA. Fourth, list any two limitations of RIA. Fifth, what is enzyme immunoassays EIA? Six, name any two enzymes commonly used in ELISA. Seven, differentiate between direct and indirect ELISA. Eight, what is the principle of competitive ELISA? Nine, write any two advantages of ELISA over RIA. 10, what is the principle of chemiluminescence in immunoassay? For 5 mark question, first explain the principle and application of radioimmunoassay, that is RIA. Second, describe the steps involved in direct, that is sandwich ELISA with a neat label diagram. Third, explain the principle and interpretation of indirect ELISA used in HIV testing. Fourth, write a detailed note on competitive ELISA and its diagnostic importance. Fifth, describe chemiluminescence immunoassay principle, enzyme use, and advantages of this. 6. Explain the Western blot technique and its role in HIV confirmation. 7. Write the principle and components of flow cytometry. 8. Enumerates application of flow cytometry in clinical immunology. 9. Discuss immunoelectron microscopy and its variant with their applications. And last 10, compare and contrast RIA, ELISA, and chemiluminescent assays in terms of principle, detection, and safety. So with this, we completed today's class about RIA and ELISA. So I hope this concept is cleared. So that's all for today's class. Thank you.